Welcome to the Mystic Podcast Cruise. I'm Jake Hart. Today's episode takes us far from the realm of the mystical and into one of the most surreal, real-world battles humanity has ever fought, one that sounds like science fiction but is disturbingly real.
I'm Zara. Deep in the jungles of Panama, a quiet war has been raging for over 50 years, not against an army, but a parasite. A relentless, flesh-eating fly that once terrorized entire continents.
Each week, over 100 million irradiated flies are dropped from planes in a stunning feat of biological warfare. It's a campaign of precision, science, and desperation.
In this deep dive, we examine how the screwworm nearly conquered the Americas and the astonishing effort to hold the line using radiation, meat factories, and an invisible wall of sterile insects.
So right now, literally as we speak, high up in the air over the jungles of Panama, it's happening.
Yeah, planes dropping millions upon millions of flies blasted with radiation.
It sounds completely unreal, like something from a weird sci-fi movie or maybe the strangest weather report ever. But it's real. It's happening now.
And it's not just, some abstract experiment. This is part of a war, a quiet war that's been going on for decades, not against people, but against this one specific, really terrifying parasite.
And that parasite is Cochleomia hominivorax.
Yeah.
That name alone, hominivorax, it literally means man-eater. We're talking about the New World screwworm fly.
And man-eater is, chillingly accurate. This isn't your average housefly. It's a parasite that specifically targets warm-blooded animals. Its whole life cycle revolves around finding wounds, laying eggs inside living flesh.
And then the larvae. They eat the host from the inside.
Exactly. They eat living tissue. The amount of suffering this one creature has caused, historically and even now when it breaks through defenses, is just immense.
But the flip side, the human response, is almost as unbelievable as the fly itself. For more than 50 years, we've been locked in this fight. It's not conventional warfare, obviously.
Not at all. It's a war fought with advanced science, incredible logistics, basically a form of biological warfare against the parasite. Breeding billions of flies, hitting them with radiation to sterilize them, and then just dropping them out of planes.
Across huge areas, too, from the southern U.S. down through Mexico, Central America, right down to Panama. And wasn't there even an incident in Africa?
Oh yeah, a major one in Libya. It highlights just how seriously this threat is taken. This whole thing, it's a story about human ingenuity, I think, staring down this horrifying natural enemy and coming up with something radical.
Radical ideas that actually worked. Incredible engineering, massive coordination, and this invisible wall of sterile flies that's still protecting millions of animals and, frankly, billions of dollars in agriculture.
Right, even today.
So our mission here is to unpack this whole incredible story. How did this fly become such a menace across the Americas?
Yeah.
How did scientists come up with this, frankly, insane-sounding idea of fighting flies with flies?
And how is this bizarre high-tech war still being fought right now, high above the ground?
Let's deep dive into this battle. To really get it, we have to start with the enemy itself. Cochleomia hominivorax, the New World screwworm fly.
Okay, so if you saw an adult fly, you might not immediately think monster. They're actually kind of striking looking. Metallic blue-green body, really bright red eyes. They usually have 3 dark stripes on their back, too.
So at first glance, maybe like a regular blowfly or something you'd see buzzing around.
Yeah, pretty much. They blend in visually with other flies you might see, but their looks are really deceptive.
How so? What makes them so fundamentally different, so dangerous compared to, say, the maggots you might unfortunately find in a dumpster?
It all comes down to what they eat, their ecological role. Most maggots, the ones people are familiar with, are decomposers. They eat dead stuff, dead animals, garbage, decaying plants. They're, nature's cleanup crew.
Okay, part of the cycle of breaking things down.
Right. But Cochleomia hominivorax, it's completely different. Its larvae, the screwworms themselves, do not feed on dead tissue. Their entire existence, their survival as a species in nature, depends completely on feeding on the living flesh of warm-blooded animals.
So no cleanup role.
Yeah.
No benefit from our perspective anyway. They are pure parasites on living things.
Exactly. And they are terrifyingly good at finding hosts. The first step is detection. These flies have an amazing sense of smell. They can detect the scent of blood, even from a tiny wound, over incredible distances.
Like across a huge ranch, they could find one injured cow.
Potentially, yes. That sensory ability makes them incredibly persistent. Any small break in the skin becomes like a flashing neon sign for them.
And that attracts the female fly.
Precisely. Once she finds a warm-blooded animal, could be a cow, a deer, a dog, even a person with any kind of wound, even just a scratch or an insect bite, she's drawn right to it. Her one mission is to lay her eggs directly in that wound.
Inside the wound itself.
Right in there, where it's moist and has nutrients, and she's incredibly prolific. A single female can lay a batch of eggs, maybe up to 400 at a time.
400 in one go.
Yep, and she can do this multiple times in her life.
Yeah.
Laying batches in different wounds or even adding to existing infestations.
That's horrifying.
Yeah.
Okay, so 400 eggs land in a wound. What happens next when they hatch?
That's where the name screwworm comes from and where the real nightmare starts. The eggs hatch fast, sometimes within hours if it's warm enough. And the larvae, these tiny worms, they get to work immediately. Feeding.
Right.
But here's the crucial, awful difference. They don't eat dead or dying tissue around the wound edge. They use their specialized mouth parts, these sharp little mandibles, to tear into and eat the healthy, living flesh.
Oh no. So they're actively making the wound bigger, eating into the good tissue.
That's exactly right. They burrow head down, feeding as they go, literally screwing themselves deeper into the flesh. That's the screwworm action. This causes the wound to get bigger and deeper incredibly fast. It's intensely painful for the animal.
And I imagine it gets infected easily too.
Absolutely. Secondary infections are common. But here's the truly vicious part. This process creates a positive feedback loop. The bigger, nastier, more infected wound, full of feeding larvae, actually attracts even more female screwworm flies.
So one infestation basically calls in reinforcements.
Okay.
More flies lay more eggs in the worsening wound, creating more larvae, which eat more flesh. It just spirals.
It's a spiral of destruction. The wound becomes this self-perpetuating engine of suffering. It gets deeper, wider, constantly reinfested. In serious cases, which happen a lot, the host animal gets incredibly weak, suffers horribly, and dies. It's a cruel, slow death.
There's really nothing good about this fly, is there?
Nothing. It doesn't help decomposition. It just exists to parasitize living animals. It's a natural enemy in the purest sense of the word.
And this wasn't some new plague. The sources say it was just a horrifying fact of life in the Americas for ages. Animals and people just lived with this.
For centuries, maybe longer. Wildlife certainly did. Indigenous peoples knew about it. Early settlers encountered it with their livestock. It was always there, a grim background threat. But then humans, kind of accidentally, made things much, much worse.
How did we do that?
Large-scale agriculture, specifically cattle ranching. When European settlers brought millions and millions of cows to the Americas, especially across the southern U.S., they essentially rolled out a welcome mat.
A massive walking buffet for screwworms. Millions of large animals, outdoors, constantly getting little nicks and scratches.
Exactly. Suddenly you had this huge density of potential hosts. Minor injuries from fences, thorny bushes, branding, castration, even tick bites, things that happen all the time on a ranch became open invitations. What had been a persistent natural problem exploded into an absolute catastrophe.
Across the southern U.S. first.
Yeah, and it spread fast. It became this source of endless suffering for livestock. Ranchers would see outbreaks just rip through their herds. Cows, sheep, goats, horses, anything warm-blooded was vulnerable. And it wasn't just farm animals. Wildlife suffered terribly too. Deer, rabbits, even birds sometimes. Agonizing deaths.
The economic hit must have been huge for ranchers, besides just the cruelty of it.
Devastating. Ranchers were forced into this constant, exhausting vigilance, riding the range every single day, checking every animal for any sign of a wound, any flies buzzing around. Trying to treat infestations was awful, cleaning out maggots from a deep wound on a struggling animal.
And newborns would be especially vulnerable, right? Like at the umbilical cord.
Extremely vulnerable. They'd have to treat the navels of newborn calves and lambs immediately with repellents, hoping to keep the flies away. But even with all that work, all that effort, they often had to watch helplessly as animals suffered and died. It was heartbreaking and economically ruinous.
So facing this nightmare, this escalating crisis, what could they even do back then? Did pesticides work at all?
You'd think so, right? That's the usual approach for insect pests. But for screwworms, traditional insecticides were almost useless when it mattered most.
Why?
Because the target, those larvae doing all the damage, were buried deep inside the living animal's flesh. You couldn't just spray the cow with enough poison to kill maggots deep in a wound without seriously harming or even killing the cow itself.
Right, you poisoned the host.
Exactly. It's like trying to cure an internal parasite by dipping the whole patient in disinfectant. It just doesn't work safely.
And you couldn't just stop the animals from getting hurt in the first place, not on a massive scale.
Impossible. You can't bubble wrap millions of cattle spread across thousands of square miles. And with the fly's incredible ability to find even tiny wounds, you couldn't stop them from reaching their targets. So you couldn't kill the larvae effectively where they were feeding, and you couldn't prevent the flies from finding hosts. It genuinely seemed hopeless. Cochleomia was winning. Conventional methods had totally failed.
That's a really bleak picture. An unstoppable parasite, widespread suffering, economic disaster, and no tools to fight back effectively. What could possibly change that?
Well, that brings us to the 1950s. And amidst all this despair, two scientists came up with an idea. An idea that honestly sounded completely nuts at first. Probably too wild for most people to even take seriously.
Okay, too wild, completely nuts. I'm listening. What was this crazy idea? Was it really about nuking the flies, like the intro hinted?
Well, nuking is maybe a bit dramatic, but radiation was the key ingredient. The core idea wasn't just destruction, though. It came from spotting a really specific weakness, a kind of Achilles heel, in the screwworm's own biology.
A vulnerability. What was it?
It was all about the mating habits of the female fly. Now remember, she can lay hundreds of eggs, multiple times. But, and this is the critical part, she only mates once. Just a single time in her whole life, which is only about 3 weeks long anyway.
Okay. So just one shot to get fertilized.
Exactly. One chance. If that one mating is successful, her eggs are viable. If it's not successful, she'll still lay eggs, maybe multiple batches, but they'll be infertile. They won't hatch. No new screwworms.
Okay, I see where this might be going.
Right. So these two scientists, Dr. Edward Nippling and Dr. Raymond Bushland, they looked at this and thought, what if we could somehow guarantee that her one mating opportunity was wasted? What if she mated with a male who couldn't fertilize her eggs?
You mean flood the environment with sterile males? So the odds would be overwhelming that she'd mate with one of them instead of a fertile wild male.
That was precisely the idea. The concept became known as the sterile insect technique, or SIT. The theory was, beautifully simple in a way. If you could raise absolutely massive numbers of male screwworm flies, sterilize them somehow without killing them or making them too weak to compete.
And then just release them. In huge numbers, way more than the wild males.
Exactly. Release them so they vastly outnumber the fertile wild males. Then, statistically, most wild females would end up mating with one of your sterile, lab-reared males. They'd waste their only reproductive chance.
And over time.
Over time, fewer and fewer fertile matings would happen. The wild population's birth rate would just plummet. In theory, if you kept the pressure up in a contained area, the entire species could literally mate itself into extinction.
That is brilliant. Using their own biology against them. But, okay, the practical side sounds insanely difficult. How on earth do you sterilize millions, or you said billions, of flies without killing them or making them too weak to even fly or attract a mate?
That was the massive scientific hurdle, and this is where sort of lucky timing came in. The 1950s, because of the Cold War and nuclear research, saw a lot of study into the effects of radiation on living things, and scientists stumbled upon something crucial.
Which was?
That specific, carefully controlled doses of radiation, like gamma rays, could damage the reproductive cells of the sperm or eggs of an organism, like an insect, while leaving the rest of the body mostly unharmed.
So the fly would still develop, emerge, fly around, act normal, but it just couldn't reproduce.
That's the key. It could still compete with wild males for mates. It would seem perfectly healthy to a female fly. But any mating would result in infertile eggs. The radiation essentially made them reproductively dead while leaving their mating behavior intact.
Wow. So the plan became figure out the exact radiation dose, figure out how to breed flies by the billion, irradiate them all safely, and then drop them out of airplanes.
Yeah, that was the audacious plan. Breed them, irradiate the pupa that turned out to be the best stage, and then release the sterile adults from aircraft over thousands of square kilometers of infested territory.
I'm just picturing the meeting where they proposed this. Okay, folks, plan A, build massive factories churning out flesh-eating maggots. Plan B, blast them with cobalt-60. Plan C, fly airplanes over Texas and just dump them out. Trust us.
It must have sounded absolutely bonkers, like something out of a pulp sci-fi magazine. But you have to remember how bad the screwworm problem was. The economic devastation, the horrific animal suffering, traditional methods were useless, people were desperate. Eventually, the authorities, the USDA, were willing to say, okay, maybe this isn't completely crazy, let's at least see if it's possible to test it.
Desperation really does drive innovation. So where did they go to prove this wasn't just, scientific fantasy? How do you test dropping irradiated flies?
They needed a controlled environment first, so scientists set up a facility in Florida, basically a pilot plant, to work out the kinks of mass rearing millions of these flies consistently. That was a huge challenge in itself.
Figuring out how to be a fly farmer on an industrial scale.
Pretty much. Once they felt they had a handle on the rearing part, they needed an isolated place for the actual release test. They chose the island of Curacao, down in the Caribbean.
Curacao, why there?
Yeah,
Isolate.
exactly. It was geographically isolated. Critically, it already had a native breeding population of screwworms, so they knew the flies could thrive there. But being an island, it was a closed system. They could release their sterile flies and see the impact on the local population without worrying about fertile wild flies constantly migrating in from elsewhere and messing up the results.
Makes sense. Okay, back in Florida, how were they actually breeding millions of flies back then? Was it like clean, high-tech labs?
High volume, yes. Clean. Maybe not the first word that comes to mind. And definitely not pleasant. They used these long, shallow trays, hundreds or thousands of them, often stacked floor to ceiling, and these trays were filled with this specific goo, the larval medium.
The stuff the maggots eat. What was in it?
It was basically designed to mimic a warm animal wound. The recipe sounds pretty disgusting, honestly. It was mostly ground beef and horse meat mixed with animal blood, milk, and eggs.
Oh, wow. The meat, blood, milk, egg slurry for worms. That sounds vile.
Yeah, not exactly appetizing. And a key detail was the temperature. They had to keep this mixture constantly warm, around body temperature, like inside a living animal. That was crucial to get the larvae to feed and grow properly, to make them healthy and competitive enough for the next steps.
Right, mimicking their natural habitat. Okay, so they're growing billions of larvae on this warm, awful-sounding meat smoothie. Then they collect the pupa, zap them with radiation, and ship them off to Curacao.
That was the process. Once the larvae were mature, they'd pupate. The pupae were collected, irradiated with a carefully calculated dose, and then flown to Curacao for release.
And on Curacao, they just started dropping them.
They started releasing them systematically. Thousands, then hundreds of thousands of these sterile flies were released from planes at regular intervals, maybe weekly or even more often. The goal was to ensure the number of sterile flies hitting the island vastly outnumbered the estimated wild, fertile flies living there.
And how did they track if it was actually working? How could they tell?
They had teams on the ground constantly monitoring. They'd inspect injured animals, looking for wounds with egg masses. The crucial step was collecting those egg masses and taking them back to a lab to see if they hatched. Were they fertile or infertile? They also trapped wild flies to analyze mating patterns.
So they were basically counting the percentage of dud eggs being laid.
Exactly. And as the weeks and months went by, the data started showing a clear trend. A higher and higher percentage of the egg masses found on the island were infertile. The wild female flies were still mating, but more and more often they were mating with the sterile males released from the plains, wasting their one shot.
So the sterile flies were successfully diluting the reproductive potential of the wild population.
Precisely. The number of fertile wild flies just couldn't keep up. The source mentions the decline was slow at first, as the ratio of sterile to fertile males shifted, but then it became dramatic. It said, suddenly they were no more. The native screwworm population on Curacao completely collapsed, eradicated, gone from the island.
Wow. From a place where they thrived, totally wiped out by this seemingly bizarre technique. That must have been an absolutely incredible moment for nippling and bushland and everyone involved. Proof.
It was huge, a monumental success. It proved the sterile insect technique wasn't just a cool theory, it was a practical, effective weapon against this devastating pest. And that success meant, as the source puts it, it was time to think much bigger.
Bigger than an island. Time to take the fight to the mainland where the real catastrophe was happening.
Exactly. Curacao proved it could work. Now it was time to unleash it on a continental scale. A full-scale eradication war was declared, starting in the U.S. This wasn't a pilot project anymore. It was a major national and later international undertaking. And that meant scaling up production massively, building what the source calls professional worm factories.
Factories designed to churn out billions of sterile flies. Billions. That number is still hard to wrap my head around. The Texas plant example really drives it home.
Oh yeah, the plant near Mission, Texas became legendary. To hit their production targets, they were aiming for something like 150 million sterile flies per week from that one facility. The amount of raw material they needed was just staggering.
Get this. 70 tons of meat and 12,000 gallons of blood every single week.
70 tons of meat and 12,000 gallons of blood per week. just to feed maggots. Pause and think about that logistical chain. Sourcing it, transporting it, processing it, it's mind-boggling.
Truly incredible. And inside the factory, it was still that same basic, gross recipe, just scaled up.
Pretty much, yeah. Highly mechanized by then, of course. Automated systems to mix and distribute the disgusting mix, as the source calls it, into those endless rows of trays, and always kept warm, precisely at that living tissue temperature, around 98.6 Fahrenheit, to maximize larval growth. Huge climate-controlled rooms full of trays of feeding larvae, an actual fly factory.
And then the pupa get irradiated.
Yep. Once the larvae pupated, the pupa were collected, put into containers, and sent through the irradiation process, usually using cobalt-60 gamma rays. The dosage control was critical enough to guarantee sterilization of every single fly, but not so much it messed up their ability to emerge, fly, find mates, and compete effectively. That technology got refined over years.
So billions of sterile flies ready to go. How did they get them distributed? I remember you mentioning some logistical hiccups.
Like the
smell.
Oh, the logistics were a constant challenge, sometimes in really weird ways. The smell anecdote is classic. Apparently, growing billions of flies on warm meat and blood creates a pretty powerful odor. And the pupa, or newly emerged flies packed for transport, well, they stink.
How bad?
So bad that, according to the source, airlines initially refused to transport the boxes. They wouldn't put them on cargo planes.
No way. That's a major problem if your whole plan relies on airplanes.
Right. It was a serious bottleneck. The solution they came up with back then sounds hilarious now. They literally had to douse the transport boxes with cologne, just spray them down to mask the smell enough for the airline crews to agree to load them.
Boxes of radioactive fly pupas smelling vaguely of cheap cologne. That's quite an image.
It really highlights the kind of practical, unexpected problems they had to solve constantly. But they pushed through. The eradication campaign moved forward, described as this slow-moving wave of biological warfare.
Where did the wave start, and which way did it move?
It kicked off in Florida, which was heavily infested, cleared the screwworm out there first. Then the battlefront moved west, clearing the southeastern states, then pushing across Texas and the rest of the U.S. southwest. Once the U.S. was declared free, the effort became international, a joint program with Mexico to push the wave south through that entire country.
And then into Central America.
Yep, from Mexico down through Guatemala, Belize, El Salvador, Honduras, Nicaragua, Costa Rica, country by country.
That must have taken years. Decades.
Decades. Absolutely. Clearing the U.S. took years. Pushing through Mexico and Central America took many more. Every mile gain required the constant production and release of billions of sterile flies, huge international coordination, massive funding, and just relentless dedication from thousands of people. Factory workers, pilots, scientists, veterinarians, ground surveillance teams.
It sounds like one of the most ambitious pest control programs ever undertaken. A true continental-scale effort.
It really was. The source rightly calls achieving eradication from the U.S., Mexico, and most of Central America an incredible victory of humanity over the horrors of nature. It saved billions in agriculture and spared untold numbers of animals from horrific suffering.
But nature pushes back. The fly wasn't completely gone from the planet, right? And you mentioned Libya.
You're right. While North and Central America were cleared, the screwworm still had a strong foothold in South America, especially in the Amazon basin. And then came 1988, a huge shock. Screwworms somehow escaped the Americas entirely and turned up in North Africa, in Libya.
Libya? How in the world did they get there? That's a massive jump.
The exact route isn't perfectly known, but the most likely way was accidental transport. Probably an infested animal shipped by sea or air, carrying live larvae or pupa that then emerged and established a population in Libya. However it happened, it was an immediate, top-level global emergency.
Why so urgent? Because it could spread across Africa.
Exactly. The nightmare scenario was the fly establishing itself in Libya and then spreading. Down the Nile Valley into Egypt and sub-Saharan Africa, maybe across North Africa. Vast regions with huge livestock populations, abundant wildlife, warm climates, and often very limited veterinary services or medical care compared to North America.
The potential for suffering.
Yeah.
You used the word incalculable earlier. That seems right.
It really does. A flesh-eating parasite running rampant in areas without the resources to fight it. It could have been a catastrophe on an unimaginable scale for both animals and potentially people. This was not a regional problem anymore. It was a potential global disaster. It needed a massive, immediate international response.
What could they do? They couldn't just build a giant fly factory in Libya overnight.
No time for that. The response had to be fast, leveraging the existing infrastructure. It became this massive international airlift operation, coordinated by organizations like the UN's Food and Agriculture Organization, FAO, in various countries.
Airlifting what? Sterile flies?
Hundreds of millions of sterile screwworm pupae were flown from the factory, which by then was operating in Mexico, all the way across the Atlantic to Libya. These were then dispersed by air over the infested areas, just like in the Americas.
And on the ground.
Simultaneously, huge international teams of veterinarians and animal health workers rushed into Libya. They fanned out across the affected regions, inspecting literally millions of animals, camels, sheep, goats, cattle, looking for any sign of infestation, treating wounds, applying larvicides where possible, trying to contain it manually while the sterile flies did their work.
And telling the local farmers why planes were dropping boxes of flies on them.
That was a huge part of it. The source specifically mentions the communication challenge. Imagine you're a farmer there, you know this horrible fly, and suddenly planes are dropping boxes of them. You'd be terrified. They had to launch major public information campaigns to explain that these flies were sterile, they were the good guys, part of the plan to get rid of the bad flies.
Don't worry, these American flesh-eating flies are here to help. A tough sell.
Extremely tough. But incredibly, the whole thing worked. The sheer scale and speed of the intervention, the sterile fly drops, the ground surveillance and treatment overwhelmed the newly established wild population before it could really take hold and spread. Within just 4 months, they declared Libya screwworm-free again.
4 months, that's astonishingly fast. Stopped a potential continental invasion in its tracks.
An amazing success story of international cooperation and rapid response using the SIT technique. But as you pointed out, this didn't solve the underlying issue. The screwworm was still thriving in South America.
Right, so after Libya, the threat was contained, but the source population in South America was still there. Why not just keep the eradication wave going south? Clear the whole hemisphere.
The sheer scale of South America, especially the Amazon rainforest, made that seem impossible, or at least impractical at the time. It was deemed just too vast, too geographically complex, too expensive, and too politically complicated to coordinate across all the different countries involved.
So, if eradication wasn't feasible for South America, but letting them come back north was unthinkable after all that effort, what was the plan?
The strategy shifted from continent-wide eradication to permanent containment. Build a wall. Not a physical wall, but a biological barrier. They decided to establish this barrier at the narrowest point of the land bridge connecting the continents, the Isthmus of Panama.
Hold the line in Panama.
Exactly. A permanent defensive line. An agreement was struck with the government of Panama. The U.S. and Mexico would provide the funding and technical expertise to build and maintain a massive sterile fly production facility right there in Panama, near the border with Colombia. Its sole purpose, to create a permanent wall of flesh, or rather sterile flies, to block any screwworms trying to migrate north from South America.
So the main battlefront moved permanently to the Panama-Colombia border zone.
That's where the continuous fight happens now. There's a dedicated facility, COPEG, the Panama United States Commission for the Eradication and Prevention of Screwworm, operating 247 deep in Panama. Still sometimes called the nuclear worm factory colloquially because of the radiation involved, though it's a modern biofacility. Its job is to produce an absolutely endless stream of sterile flies.
Is it still that awful meat and blood recipe? Or have things improved?
The core principle is the same: grow larvae on something mimicking worm flesh, but the tech is way more advanced now. Instead of handling raw meat, they use what the source calls a brown protein sludge. It's basically powdered blood, milk, and eggs mixed with water and other nutrients formulated into a consistent slurry.
Sounds marginally better?
Maybe slightly less gruesome to imagine, but probably still smells awful. This sludge is piped automatically into those long trays in huge climate-controlled rearing rooms, kept precisely at that 37 degrees Celsius, 98.6 Fahrenheit living tissue temperature. It's highly optimized for mass production.
And the sterilization process?
Yeah.
Still radiation?
Still radiation, yes. Cobalt-60 is often still the source. The pupae are collected, packaged, and moved through highly controlled irradiation chambers. The dosage is incredibly precise now, fine-tuned over decades. The goal is always the same: sterilize them completely without affecting their mating competitiveness. Make flies that act normal but are dead inside, reproductively speaking.
And these flies form the invisible wall. How does that work in practice? How do they deploy them?
It's a continuous aerial operation. Every week, massive numbers, the source says 100 million or more, are loaded onto specialized aircraft. These planes have sophisticated dispersal machines that gently release the chilled, inactive flies at a controlled rate as they fly specific routes.
So they're basically seeding the border zone with sterile flies constantly.
Exactly. It's described as a finely tuned balance of drop rate, speed, and altitude. The flight paths are meticulously planned, like a grid, often separated by about 1.6 kilometers or a mile. This creates overlapping zones of sterile fly
Oh,
release.
choreographed aerial ballet is the phrase used.
It's a good description. This constant, precise rain of sterile flies creates that dense, invisible barrier along the Panama-Colombia border. Any fertile, wild female fly trying to move north from South America has an extremely high probability of encountering and mating with one of these sterile males first, effectively stopping her lineage right there.
It's a permanent defense, but it must need constant checking to make sure it's holding.
Absolutely critical. Surveillance is ongoing. Teams on the ground, often working in incredibly remote and difficult jungle terrain along the border, are constantly checking livestock and monitoring wildlife traps. They're looking for any sign of a wound, and especially for screwworm larvae. Any larvae found are collected and sent for analysis to confirm if they're wild fertile flies indicating a breach or just sterile ones from the drops. This is the early warning system.
It sounds incredibly robust.
Mm.
Decades of refinement, billions of flies, constant monitoring, like a fortress.
Yeah.
But you said at the beginning, unfortunately right now, the wall is failing.
Yeah, that's the really tough part of the story right now. Despite all that incredible effort and technology, that fortress isn't completely impenetrable. The war against cochleomia is absolutely not over.
What happened? Where did it fail?
There have been worrying signs. Back in 2016, there was that outbreak in the Florida Keys. Screwworms just suddenly appeared, thousands of miles north of the barrier. It was a nightmare for the endangered key deer population there, horrible wounds suddenly appearing.
How did they get all the way to Florida? Did they sneak past the Panama Wall?
It's still debated exactly how that happened. Could it have been a failure further south that went undetected for a while? Or maybe a separate introduction, like on a boat or smuggled animal? We don't know for sure. But the system did react quickly.
They flew in sterile flies from Panama.
Immediately. Millions were airlifted from the Panama factory and dropped over the Keys to contain and eradicate that specific outbreak. The source says it was at least contained within a few months, stopping it from spreading to mainland Florida. So a scary breach, but the response worked.
Okay, so that was contained. But you mentioned a current crisis being more severe.
Yes. Unfortunately, starting in late 2023, there seems to have been a much more significant failure of the containment wall in Panama itself. Screwworms managed to push north from Colombia through or around the barrier zone on a larger scale, and they spread fast.
So where?
The source calls it a parasitic firestorm on speed. Infestations rapidly spread across Panama into Costa Rica countries that had been screwworm-free for years. And worryingly, recent reports say they've even been detected back in parts of southern Mexico.
Mexico. Areas that were cleared decades ago in that huge eradication campaign. They're back.
It appears so, in some areas. This is a genuine biological emergency unfolding right now. The impact on livestock, wildlife, and potentially even human health in those regions is serious. The Panama factory, Copeg, is running flat out, producing at maximum capacity to try and fight back.
Pushing sterile flies into all these new outbreak zones.
Yeah, exactly. Trying to reestablish containment lines, push the infestation back south again. But it's a massive, difficult, and expensive fight.
Yeah.
And the source is clear. It's not clear when it will be over.
Wow. So after all this time, all this science, all this effort, the screwworm is still fighting back hard. It's not history. It's happening now.
It absolutely is. It's a powerful reminder that these kinds of biological battles are never truly static. Environments change, parasites adapt, maybe funding fluctuates, vigilance slips, and the enemy can surge back. Maintaining these victories requires constant effort and resources.
But there's still hope, right? The source suggests the war can eventually be won. We've seen eradication work before.
Yes, there's definitely hope. Eradication from huge areas was achieved. The ultimate dream is global eradication, though that's an immense challenge. But other devastating parasites and diseases have been wiped out, or nearly wiped out. It's possible. It just requires unwavering global commitment.
So if you happen to be traveling in Central America, maybe near that Panama-Colombia border,
Yeah.
and you look up and see a low-flying plane flying a grid pattern,
You're probably not watching crop dusting. You might be witnessing a sortie in this incredibly long, strange, and vital war. A battle fought with radiation and billions upon billions of sterile flies.
A war we simply cannot afford to lose, given what happens when this fly gets loose.
And the whole story, really, from the pure horror of the fly to the sheer audacity of the solution, it's just an amazing example of human ingenuity, isn't it? Think about proposing that idea in the 50s. Using radiation to sterilize flies and drop them from planes, it sounds crazy even now.
It really does. It took people looking at a seemingly hopeless problem and seeing a completely different angle, a weird biological trick. That kind of thinking
Is incredibly valuable. Seeing solutions where everyone else just sees a dead end, even if the solution sounds utterly bizarre at first. Understanding the system deeply enough to find that leverage point.
And it also makes you realize these huge, almost invisible battles are being fought all the time. Not with soldiers, but with science, logistics, dedication,
Yeah.
protecting our food supply, our environment, sometimes our health, from threats we barely even know exist. That invisible wall of flies over Panama is just one example.
It really puts things in perspective. We worry about big headlines, but these constant grinding wars against devastating natural threats demand so much unseen effort and innovation just to maintain the status quo, just to keep things from getting much, much worse.
So let's recap this incredible journey. We started with the terrifying reality of this flesh-eating fly, Cochleomia hominivirax, and how it ravaged the Americas.
And the failure of all the old methods, leading to this radical, almost unbelievable idea of sterile insect technique nuking flies, essentially.
The proof of concept success on Curacao, showing it could actually work.
Followed by the monumental, decades-long continental eradication campaign across North and Central America, the worm factories, the cologne-sprayed boxes, the slow wave of progress.
Then the sudden global emergency when it escaped to Libya, and the incredibly rapid, successful international response to stamp it out there.
Leading to the strategic shift, building the permanent biological wall in Panama with the modern fly factory and the constant aerial ballet of sterile fly drops.
And finally, the sobering reality of today. The wall isn't perfect, the screwworm is pushing back, and the war is very much ongoing. A real biological emergency requiring maximum effort right now.
It's a story of nature's resilience meeting humanity's relentless ingenuity.
A scientific marathon against a truly horrible adversary.
And that brings us to the end of this deep dive, tracing this extraordinary battle across continents and decades.
Which leaves us with our final thought for you. Considering this unique, decades-long war against a single parasite fought with radiation, mass breeding, and an invisible aerial barrier, what does the ongoing struggle in Panama teach us about confronting persistent natural threats? What does it reveal about the limits, but also the incredible possibilities of large-scale biological control? And maybe, what does it suggest about all the other unseen, essential battles being waged constantly, just beyond our view to protect the world we live in?
There's always more beneath the surface, or sometimes flying high above it. Fascinating stuff to think about.
Thank you for joining us on this episode of Mystic Podcast Cruise. If you enjoyed today's story, give this video a thumbs up and share it with others who love uncovering life's mysteries. Be sure to subscribe to our channel and hit the notification bell so you never miss an episode. Your support helps us continue bringing you captivating stories about the unknown, the mystical, and the extraordinary. Until next time, stay curious, stay open, and keep cruising through the mysteries of life with us here on Mystic Podcast Cruise.